My name is Phyllis, and today, together with Isabella Valentin, on behalf of the ESRS Joint Subcommittee, I would like to welcome you to the inaugural session of the ESRS Joint Seminar Series. This new series aims to bring together sleep and circadian researchers from across America and Europe. Each seminar will feature one European and one American scientist offering a distinct perspective on a shared topic and will be held quarterly, aligning with the season-specific theme. All of our attendees have been invited to send in questions for today's speakers, and we will leave some time at the end of the lectures for an open discussion.
We are kicking off 2025 with our spring seminar on circadian rhythms. We have two fantastic speakers today. Merrow will present on the concept of circadian organization, the key to effective circadian medicine, and Phyllis will be discussing circadian medicine insights from the clinic. First up is Dr. Merrow. Dr. Merrow is a chronobiologist known for her research on circadian rhythms in humans and also on model organisms. She's a professor at the Ludwig Maximilian University Munich. Please welcome Dr. Merrow. The stage is yours.
Thank you so much. It's really a pleasure to be invited to be part of this series. Let's see if I can get this up now. Yes, very good. How does that look for you? Is everything okay?
Great.
Good? Okay. So I'm going to talk today about circadian organization. And my task is to give a little bit of a basic background on some key features of circadian biology that I think, and that Phyllis agrees, I think are important to understand the concept of circadian medicine. And I think in the end, I'm a little bit of the pessimist, although a hopeful pessimist. And Phyllis is probably more the optimist, also hopeful and really full of activity and really implementing lots of exciting things. So let's see, to get you on board to understand a little bit about the circadian clock, how do we recognize the circadian clock in the first place? So this is a graph that you will commonly see when you are looking at circadian data describing circadian activity. And what you see here is you see that you see 1, 2, 3, 4, 5, 6, 7 days listed from top to bottom in this case, and it could be activity level, gene expression, some quantity that you're measuring over the course of the day, and then you see the hours of the day down below on the x-axis, so it's 0 to 24. So it's a single day across the bottom, and then each day going from top to bottom of a week. And what you see here is a cartoon example of data that is showing something that spontaneously has a circa 24-hour rhythm, about once a day. That's longer than 24 hours. You see that by graphing it this way, you automatically get the idea that it's longer than 24 hours. And here you see a cartoon example of something that's shorter than 24 hours. It's spontaneously rhythmic in constant conditions, either in constant darkness or in constant light. And this is the observation that the pioneers of the field were grappling with and so amazed by, to see these spontaneous rhythms come out in all kinds of organisms. Okay, nothing's advancing. There we go. If you look at circadian clocks or this daily activity or gene expression in the context of a normal light-dark cycle, as we see every day, a 24-hour light-dark cycle, you'll see something interesting happen. You'll see them gradually come into some kind of a stable phase relationship with the light-dark cycle. In this case, or as we say, the Zeitgeber cycle. Zeitgeber is German for time giver, and their circadian clocks synchronize or entrain to a huge variety of these signals from the environment that are reliable predictors of the time of day. The strongest one, the one that's most commonly used and is most dominant, is the light-dark cycle. So you see this comes in and it finds a stable phase relationship here. And then you have something that's rhythmic exactly once per day. So circadian clocks free run, and they also synchronize or entrain to Zeitgeber cycles. This is relevant for this audience here because it also extends to humans, of course. And this is a beautiful example from Jürgen Aschoff, who did pioneering studies on the human circadian clock. And what you see here is a 30-day experiment. You see the same setup here with 0 to 30 days. And then here you see 1, 2, 3 days across the x-axis. And this person went into the time isolated, the temporal isolation environment here, and they could see what was going on outside for 7 days. And then they were in a free running, constant condition for 2 weeks. And then again, the door opened and they could see what was going on outside. So in this one graph, this one 30-day experiment on a single human, you see the concept of entrainment or synchronization, and then this free running rhythm in constant conditions, and again, entrainment. So this is what humans do. When I saw this data, there were about 400 subjects that went through this temporal isolation environment that Aschoff and Veva built. When I saw this data and graphed it out, I couldn't believe, I said, that would never be me. But of course, all of us would do this if we were put into temporal isolation or something very similar to it. There are a few exceptional phenotypes that come out, but this is the general pattern that you see when you put someone into such a protocol. So we also have a circadian rhythm that's regulating our sleep and wake cycle. Entrainment occurs, this is what I've just shown you, and I think it should be obvious by now. Entrainment occurs when a free-running circadian rhythm fits itself into a Zeitgeber cycle. So it's non-24, it's almost 24 hours, but then it has to fit itself into a Zeitgeber cycle. And what you also see here is that in this case, it takes a little bit of time to come in to find the right phase relationship, a stable phase relationship with the Zeitgeber cycle. So the first concept that I wanted to mention then is that circadian clocks can entrain faster or slower depending on their own robustness, sort of their own momentum, and the Zeitgeber strength of in their environment. And you can see this, you can imagine this. This occurs because all of our endogenous circadian clocks are not identical. Some people have high amplitude clocks, some people have low amplitude clocks, some people have shorter, and some people have longer circadian rhythms. Similarly, Zeitgebers are varied. They vary according to our life cycles. Some of us are outside all the time. Some of us have very dark nights. Some of us have gray or dim light nights, and they come in all forms also. And this results in a variety of phase relationships of the sleep-wake behavior to the light-dark cycle that we expose ourselves to. And this can be described as chronotype and as the distribution of chronotypes. So this combination of the biological oscillator that is the circadian clock and the physical Zeitgeber, which is the light-dark cycle as I've described it, can lead to this distribution of chronotypes, distribution of phase relationships. This example here is taken from the Munich Chronotype Questionnaire, and in this case, the x-axis is showing the mid-sleep time on free days with a correction for the sleep loss that occurs on working days. And you see here the number of subjects on the y-axis. And you can see almost a near Gaussian distribution with the midpoint being around 4 in the morning, meaning that if someone sleeps for 8 hours a day, the average person is going to be going to bed at midnight and getting up at 8 in the morning. And I think that there are a couple take-home messages here. I mean, one is to bring home the point of this wonderful myriad of combinations of individual oscillators and Zeitgeber cycles that leads to this curve. The other is that you hear a lot about early chronotypes and late chronotypes, and this is interesting and this is not trivial, but indeed most of us actually are somewhere in here. So the vast majority of people are, within maybe 4 hours of each other. And so there is a dominant chronotype that you could use to target circadian medicine for normal people. I just wanted to point out that the chronotype questionnaire was validated using melatonin. So, and I wanted to point that out because Phyllis will undoubtedly come back to that later, and because this an audience of sleep researchers will be familiar with melatonin, which has its onset before sleep sometime before sleep onset, and is sort of is expressed during sleep, and then is suppressed again or degraded when with light exposure. And you can use dim light melatonin onset as an experimental method to determine circadian phase. It's not great for routine use in my mind because it's rather tedious and hard to do and a bit expensive, and but maybe those challenges are being overcome. So dim light melatonin onset is one of the measures that we use for circadian phase in humans. It's probably the best measure for circadian phase in humans at this point. So I've just talked a little bit about the chronotype distribution. So a late chronotype would have, this is not really timing of behavior. The timing of some phenotype would be relatively late in a late chronotype compared to an early chronotype. That's obvious. And a moderate chronotype, it would be somewhere in the middle. Similarly, you can modify chronotype by modifying the Zeitgeber strength, by modifying the light environment, essentially. So if you take a moderate chronotype, put them into a very low light environment, a low amplitude light dark cycle, you can push their chronotype to a later phase. If you take them and put them into a very high amplitude light dark cycle, so bright light all day and really dark nights, you can push their chronotype a little bit earlier. So just examples of the kinds of things that actually come into play when we're talking about chronotype. The concept here is that circadian clocks entrain earlier or later, depending on Zeitgeber strength and according to the Zeitgeber structure. So what does this have to do with the topic of circadian organization? Well, now we can talk about how the clock is put together. And at some level, the clock must work in this way. I've shown you this free running rhythm. And so that implies that there is an oscillator that's generating a rhythm somewhere. It's directing outputs at least the sleep-wake behavior and at least dim light melatonin onset from what I've told you so far. In addition, I've shown you that the clock entrains using Zeitgeber, so there has to be an input pathway coming into the oscillator for that. So that's sort of a cartoon version of how we can envision how the clock works. Furthermore, we know from some remarkable mutant screens done in the 90s by a variety of colleagues that the clock in animals and in plants now and fungi and is regulated by a transcription translation feedback loop. In mammals, it involves period and cryptochrome and clock and BMAL, which are activators, which then activate their own repressors. It's not so simple. The molecular clock, it turns out, is itself a collection of oscillators. There's the E-box feedback loop, and then there are these auxiliary loops, which actually also have quite a robust function on the clock also. We don't talk about them so much, but they're pretty important. So the molecular clock looks like this, and it turns out that it's operating in each cell. And so this diagram starts to approach the idea of circadian organization. So within each cell, you have a multi-oscillator molecular mechanism. The cells obviously are somehow coordinating with their neighbors. Achim Kramer has recently done some nice work with this to show that even just garden variety cells, not cells that are specially designed to communicate with themselves, can actually synchronize using secreted factors like TGF-beta in his case. Organs come together to form their own clocks, and I want to show you a bit of data on that next. And then, of course, you have at the organism level. And so you can start to see now that the problem of circadian organization of this mess is really not trivial. I wanted to just show you the data on the organs because I think it's extremely important and fascinating. This is a really fantastic experiment that was done by Zhang et al. In the Hoganesh lab, where he took, this has been actually repeated now by a number of other groups. It's just really fascinating. He took tissue from animals over 24 hours, from mice over 24 hours, and looked at the circadian transcriptome. And you can see that that there are fewer genes that are regulated in some tissues. In this case, 3% of the genes in the hypothalamus are regulated, whereas about 20% of the genes are regulated by the circadian clock in the liver. So different tissues, different amounts of genes that are regulated. And you can see that there are more genes at certain times of day in some tissues than other times of day. And this is sort of, you can look at these little clock faces here to get an idea of how different they can be. Some are facing the predominant phases go in this direction, and some that go in up and down in other directions, and it's just a fantastic demonstration of how there are cell and tissue specific circadian clocks, and they will each have their own zeitgebers. Forget about light for now, and their own characteristics, their own robustness and connectivity to the cells around them. Cells have to entrain too, and this is really an interesting challenge to understand. So the problem then of internal synchrony. Remember, I showed you this data, and I said that this value, this mid-sleep time, was sleep corrected for the sleep timing on work days. So that led to the description of an important concept called social jet lag, and this refers to the fact that the majority of us use an alarm clock on work days, and we therefore do two things. We sleep deprive ourselves a little bit or a lot on work days, and we also expose ourselves to exotic light cycles, which our circadian clock is maybe not expecting. Maybe our circadian clock is expecting that we should be sleeping, but we're getting up and exposing ourselves to light, and maybe even going outside and commuting to work in the bright light. And this has been quantified by Till Roneberg and myself, and as the difference between the sleep timing on free days and work days. And you can see that late chronotypes, as expected, have a very large social jet lag, and early chronotypes can have even none, or even a negative social jet lag. They might be jet lagged on the weekends and not on weekdays. And this basically leads to weekly cycles of moving between light environments. And Juliet Zerbini has looked at this to see. Now, I showed you that in different tissues you can have clocks that are set at slightly different times. And here you see that the dim light melatonin onset on free days moves later from weekdays to weekends. Just over those 2 days, it moves later just from disrupting your clock by sleeping at different times, especially in the late chronotypes, whereas in the early chronotypes, it stays pretty much the same as would be expected because they're probably sleeping at the same time throughout. So this is a physiological demonstration that moving your sleep between work and free days actually is doing something in the brain, which has effects and is percolating throughout the body. In the 2000s, Mike Menaker did some really key experiments where he actually demonstrated that different tissues resynchronize to a jet lag at different rates. And here's an updated version of that. So you'll hear from Phyllis about some of the peripheral clocks, and especially the liver, which is very interesting because we entrain the liver with feeding time. But here's an interesting experiment that follows up on the Menaker experiments, which demonstrates beautifully that the SCN shifts already. It's already phase shifted in an experiment in the lab with mice by the second day. But if you look at peripheral tissues, you can see that they have not come near to recovering by the second day. They take the full 9 days to recover here for the spleen, the full 9 days to recover here, or they take up to 9 days to recover for the thymus in the BAL F2 mice. But the C57 mice, they still haven't recovered completely from a 6-hour jet lag by the 9th day. And on the second day, they're nowhere near adjusted to the new time. And so this demonstrates the concept of the tissue specificity of each clock and Zeitgeber combination, what Zeitgebers are helping it re-entrain. And it also demonstrates that you can have differences due to genotype, due to genetic background. I just want to leave my last slide with saying that I guess that Phyllis will also refer to this, that of course the problem, the solution to internal time and synchrony is to be able to measure internal time. And of course, there are several groups that are working on describing molecular biomarkers of the clock. This is from the Kramer group, but there is also the Rosemary Brown group in Chicago, and I believe Derek Jan Dijk is working on this in Surrey, and I hope that Phyllis will fill in the rest of the groups working on it. It's a fantastic idea. I worry about whether it captures enough to tell us about internal synchrony. So that's my background for the problem of circadian organization and leading to circadian medicine. And I'll leave you with these last take-home messages. The timing of behavior could be mismatched relative to the timing of physiology. I hope that I've convinced you of that. I hope I've also convinced you that we don't know enough about circadian organization, that circadian medicine will depend on an understanding of internal time, that sleep timing, melatonin levels, I didn't talk about body temperature, but that's in there too. That's also clock regulated. They won't be directly indicative of the whole variety of internal times of different tissues, different cells, different internally circulating Zeitgebers. And I think one of the puzzles that I'm really thinking about is how best to describe the parameter space of internal times. So if you think of this problem of circadian organization and synchrony, what defines good synchrony? How much flexibility is tolerated for a healthy circadian clock? Do late chronotypes and early chronotypes, do they have the same phase relationship of all of their internal clocks, or are they completely different? And is that all fine? I think there's a lot to discover, and I now want to turn it over to Phyllis so that we can start the debate.
Thank you very much, Dr. Merrow, for such an enlightening presentation and these important questions raised. Now, it is my great pleasure to introduce Dr. Phyllis. Dr. Z is a neurologist and sleep medicine specialist, widely recognized for research on circadian rhythms and sleep disorders. Dr. Z is the director of the Center for Circadian and Sleep Medicine and the chief of sleep medicine at the Northwestern University. Please welcome.
Thank you very much. And I'd like to thank the ESRS and the SRS for putting together, sponsoring this joint series. And of course, when I was asked whether I would speak with Dr. Martha Merrow, I think I immediately said, of course, yes. So it's a great pleasure, Martha, to be speaking with you on this topic. So Dr. Merrow gave a really brilliant, I think, introduction, but of the complexity of circadian organization, and also helped define some of the really, I think, most important concepts of what do we mean by the term circadian? What do we even mean by the, misalignment? Because I think clinically, we tend to use this very loosely. And the concept of diurnal versus circadian, we think anything that has rhythm is circadian at this point from a clinical standpoint. So what I thought I would do, I may not be able to cover everything that Dr. Merrow thought that maybe I would cover. I thought I'd just give you some introduction, but also two clinical examples that are very different from each other as to how broadly we can apply these basic principles of circadian organization to the clinic. But before I do that, I want to let you know that for those of you who don't know, that I do come from a basic science background, that my mentors, Fred Turek and so forth, we're also their mentors with Colin Pittendridge and Michael Menaker. So I'm a grandchild, as Mike used to say, of that family. And I did start my work with hamsters and mice and also rats. And then somehow in the 1990s, I said, I'm going to start running around in this and translating this into the human research. I just want to give you that background, that indeed bridging this is really, really important. And understanding the basic principles, as Martha mentioned, is really key to really good clinical science. Dr. Merrow already mentioned, the organization. I just wanted to point out that indeed, we also have to think about the sleep-wake cycle. And now it's beginning to show that there's this bidirectional interactions. The clock helps, of course, determine the timing, quality of sleep, but the timing of sleep, but also the quality of sleep seems to be able to also feed back either directly or indirectly to the central clock. And so it's really this bidirectional relationship. We speak about that. And the same things that affect the circadian clock system, such as light, will also affect sleep directly. So there are all these indirect effects of light that are not necessarily classical circadian effects that also affect our behavior. And it's really the synchrony, of course, between these peripheral clocks and the central clocks that becomes, I think, the topic of why any alteration in the circadian organization can affect the quality of sleep, but also predispose one to diseases. So let me just go back and say, well, what is a circadian disorder then, along the lines of Dr. Merrow? Like, let's define some of these. I don't think I actually have a good definition, but we thought it could be broadly thought about a disturbance, dysregulation of the circadian system, not anything else of the circadian system that could be due to external force, environmental, behavioral issues, misalignment, that then results in a impairment of a function of the body's normal structure. So it's very, very broad. By and large, what we've looked at, most of it is related, as Dr. Martha Merrow mentioned, circadian misalignment. But there are many diseases that we are seeing now that is an issue of circadian amplitude as well, especially neurodegenerative disorders. And so in health, we have robust rhythms. There's a little bit of this, some are a little bit later, some are a little bit earlier, but within what Dr. Merrow said, between the 3 or 4 hour region. And then when you have disease, you have lower in the amplitude, but also a misalignment of rhythms. So these are pretty much the basics of where we can think about when we study these disorders, where they may be. Just another one, just saying you can misalign your internal, your SCN with your peripheral clocks as well, whether it's light, social activities, behavior, which we have studied a lot with, and also the timing of sleep and wake, and that can cause circadian and sleep disruptions. I would say it together because it's hard to disrupt circadian rhythms without disrupting sleep. And as Dr. Merrow mentioned, misalignment between tissues. Now there's this whole host of disorders. Every week there's another disorder that if we studied it, you will find that there is that circadian dysregulation could be playing a role in this. Of course, the sleep-wake disorders are the ones that we think about mostly clinically, but there's our lab has more kind of neurological disorders, a lot of cardiovascular, metabolic disorders. I'll show you a few examples of that, and allergic immunological disorders. I'll show you an example of critical care, and the list really, really goes on. So those of you who are interested in translating your basic science work to the clinic, it's a totally wide open area. We're just touching the tip of the iceberg here. And of course, if you're not well, if you have these disorders, that in turn, in a bidirectional manner, can affect the health of the circadian system itself. So you can see how in some ways, so we're going to start with the first example, which everyone is very well aware of, which is the circadian rhythm sleep-wake disorders. That is how clinically we tend to think about, when we think of circadian disorders, we think of the sleep-wake disorders because we can diagnose these, we can bill for these clinically. And so I'll talk a little bit about delayed sleep phase and probably a little bit about this non-24-hour because they're related. Why? It's an example of how a clinical problem in which we don't understand the pathobiology that the basic science can help us inform that and how we can use that to understand why these people have this problem and then effectively in a personalized manner be able to treat those. So of course we can think about it as genetic. We do know there are mutations, familial and sporadic mutations of the core clock genes like the period gene that can result in a long circadian period, for example, and that could be a predisposition to developing delayed sleep phase. But of course, there are all these other behaviors, your sleep-wake cycle, your feeding cycles, as we know, can also do that. And of course, photoreception. So that's what I will focus on, just one example of our work. This is a patient, 21-year-old female with difficulty falling asleep since 5th grade. During the pandemic, I started working from studying from home. Bedtime was delayed even further. Typically takes her 2 hours to fall asleep, sets her alarm to about 8 o'clock, wakes up a little bit during the night, but usually gets right back to sleep. On weekends, she's a little bit later, just like that later chronotype that Dr. Merrow spoke about. But she really would like to wake up mainly between 10 and 11 a.m. If she doesn't, she's tired all morning, consumes a lot of coffee, and is thought to have Asperger's syndrome, kind of in the spectrum of disorders. So the question sounds like somebody could have just insomnia disorder, or could they have a circadian rhythm sleep-wake disorder, like a delayed sleep phase? Well, if you look at their actigraphy data, it's hard to tell, right? She's sleeping late, she's going to bed late, and she's waking up late. And she's got she takes off her active watch here often. Well, yes, I'm going to talk about melatonin. And so if we sample melatonin, saliva in home, we do this all at home, you see that normally we would be expecting melatonin to be rising, the dim light melatonin to be rising somewhere between 9, 10, maybe even 11 p.m. In this individual, it's much, much later, as you can see. So I would say this person has a circadian rhythm sleep-wake disorder of the delayed sleep phase type. So why? Well, we don't understand the pathophysiology well, but it could be the light input pathway, right? And this is where basic science informs us. What about the melanopsins? There's something unusual about their light sensitivity, or it's just, as we thought, the timing of light. So maybe they're getting too much light at night and not enough light in the morning. Sure, that can cause a delay in the circadian rhythm. As we show that if you live in a very bright light-dark cycle, you tend to advance your rhythms a bit, the robustness of that. Or could it be sensitivity? So we focus a little bit on that. That's just one example of how this basic science is really informing us about what to do. So the Australia group, Watson Hank's group, they looked at patients with delayed sleep phase wake disorder, and they exposed them to light right here in the middle, here in the kind of probably in the evening hours over here. And what they found was that those with delayed sleep phase had increased sensitivity to this evening light. So that could be one of the reasons why their phase delayed, higher sensitivity. You can see here that the phase shifts were larger, and also in those with delayed sleep phase in the red. And also if you look at melatonin suppression, the same amount of light suppressed melatonin more in the delayed sleep phase group, and you can see that from the pupil size. There's something going on there. But that could be one explanation. Now, at Northwestern, Dr. Abbott and I, what we thought was, okay, we're going to take our patients, we're going to see whether that is the case as well. And we did what we call a PIPR, which is the post-elimination response. And we gave them here 5 minutes of dark, 5 minutes of red light, and then we gave them this kind of blue enriched light for about 5 minutes. And what we found was actually the opposite. We found that the individuals with delayed sleep phase disorder, they were over here. They were dilating, their pupils were dilating faster. So almost they were less sensitive to light. But this is during the day. Okay, this is mainly, we did it during the day, not in the evening. And so we looked at these patients with delayed sleep phase. Here at the top, you can see from the actigraphy records. And this is a delayed sleep phase person who intermittently becomes non-24-hour. They're sighted individuals. And what we found was that if you look at the PIPR, the post-illumination pupillary reflex, you find that there's a decreased sensitivity in the delayed sleep phase. And we look at these non-24-hour types, they were driving this. They were even less sensitive. And the later their sleep time, the less sensitive they were to light. So we thought we could develop this eventually as a diagnostic tool. Now, we have another problem clinically that about 50% of people with delayed sleep phase, they don't have a late dim light melatonin onset. So then, do they have a circadian disorder? They meet the criteria for circadian rhythm sleep-wake disorder, or do they not? And so we call them non-circadian and behavioral delayed sleep phase disorder. So Dr. Abbott and I thought, well, maybe we should think about them more as a type 1, where their melatonin, Dilamo, is delayed relative, and they have a normal phase angle, right? As you would expect, a 2 or 3 hour phase angle with bedtime or with sleep time. And they're type 2, like diabetes, right? Or narcolepsy, type 1, type 2, where the circadian central timing system appears to be intact or normal phase, but their sleep time, the phase angle, is really, really wide. So we don't understand the pathobiology of it. So therefore, I don't think it could be necessarily non-circadian. Maybe something's going on with those peripheral clocks, right? Or the homeostatic system and their interaction. So this has resulted in a new R01 study here at Northwestern to really try to understand how do these different subtypes of delayed sleep phase appear, and could it be explained by a light sensitivity mechanism? Some have high light sensitivity, some have low light sensitivity, and we're going to be looking at this so hopefully in a few years we'll be able to talk to you a little bit more about that. We have another study going on with Ken Wright, another new funded. This is 2 funded R01s on the pathobiology of delayed sleep phase disorder, and there's been really none for the last 15 or so years. So you can see what I want to point out here is that I don't want to underestimate that the modern environment, the electrical lighting and all of that could be affecting these patients in the context of vulnerability, where they may have a long circadian period, they may have high sensitivity to light, for example, or low sensitivity to light, as well as homeostatic mechanisms. So the last one, which in the last few minutes, is the totally opposite. So we have the classical sleep-wake disorders. We're trying to understand the pathobiology, and we go all the way to the critical care unit, to the ICU. So this is a 70-year-old woman who presented to the emergency room with an alteration in mental status and fever. She had low blood pressure, high temperature, elevated, inflammatory markers like a white count. So she was in septic shock. She was also not breathing well. She was also hypoxemic. Her oxygen was very, very low. She was admitted to the ICU, intubated, ventilated mechanically, put on antibiotics, and continuous feeding after 3 days. After 3 days of this, she was on continuous feeding, immobilization. We don't let people move because they may scratch themselves, they may pull their tubes out. And after, even 6 days in the ICU, she was delirious in the afternoon, in the evening, sleeps on and off all day, and she was still intubated. It was very difficult to get her off of this thing. So we had a watch with actigraphy. We had a study here. You can see, look at the light levels. They're just there all the time. There's really no amplitude to day and night. and movement was very low amplitude across the 24 hours. So the rhythms were very crushed, at least the rhythms that we could monitor. So this then led Dr. Matthew Moss, who's a neurointensivist, to really conduct several studies. One, to officially look at light levels in the ICU. You can see overall, it's not that there's too much light at night, there's not enough light during the day. So we can intervene if we wanted to do that. And if you look at melatonin, because they had we could sample melatonin as much as we wanted to because we had access to arterial lines. And you can see that compared to the green, which is the age-matched controls in the hospital, the patients in the intensive care unit, their phase of their melatonin, their melatonin onset was spread across almost 18 hours. And the same thing if you looked at the urine 6-phosphate melatonin, as you see that in the B panel. He also then he did many other things, but he also looked at core clock genes, and you can see that whether you look at BMAL, this is the ICU, pretty flat all over, very flat, timeless, period. Almost all core clock genes, very flat rhythmicity. Doesn't mean that the clock isn't ticking, just means that it's masked perhaps, at least because we're only looking at peripheral rhythms, we're looking at blood. So this gives us the opportunity, which we're doing right now, to try to change that. I mean, can we target central peripheral rhythms, for example? Can we harness the physiological data that we're getting? And Dr. Merrow and I have collaborated in the past on trying to do this, harness this data from the physiological continuous monitoring data that we have to better understand the dysregulation and to say, what can we actually do to help improve rhythmicity in the ICU, but more importantly, when they leave the ICU? This is a study by John Haganash, this group, which is really brilliant. It's a preliminary study, small sample size, where they just took the feeding, continuous feeding versus time-restricted feeding in people who are getting, I think this was stem cell transplant. And you can see very nicely that those who had daytime restricted feeding did better, right? They did better and substantially better. So this is just a pilot study, but it gives us hope that we can actually intervene in this thing. So coming back 180 degrees, almost 360, we talk about basic science, the basic principles, translational chronobiology research, and then taking that all the way to the clinic, where we can use these principles to diagnose and treat and improve health outcomes, and vice versa. I think the clinic informs the basic science to the questions that we may want to ask. And I think this is really, really important, this collaboration between all of us. The field is totally open. You can see New York Times is interested in this. They did a whole it was the front page of this article of theirs. It's relevant to aging, metabolic activity, and really almost anything you can think of at this point. So what I think the future may lie is that we will be developing circadian diagnostic laboratories, I hope. And I agree with Martha that maybe these time signatures and things that we may have, it's a great idea and it may be very useful, but we don't know what we're actually looking at because it is from blood. It's still peripheral. We don't have really great markers in central rhythm per se, although these peripheral things that we're looking at do correlate very well with melatonin, the timing of melatonin, but that's under very healthy conditions. We have tested under patients who don't have or are not healthy, for example. Clinical trials, we don't have hardly any of these in circadian medicine. We absolutely have to begin to do this. Education training of clinicians. So Martha, thank you so much. I think this is so important to get these basic principles out. What do we mean by these words? And also the complexity of the circadian organization, and to understand what are we looking at? What level of the organization are we actually looking at? Finally, for us to really make this a field, we need to get recognition by the boards of medicine, not just sleep, and requires the collaboration. All of you are listening to us, watching us, whether you're a trainee or not. And finally, I'd like to thank my group that has done all of this work and much, much more. So, and also the funding agencies. Thank you.
So thank you both, Dr. Merrow and Dr. Z, for these amazing, excellent talks. And now we move for some time for our questions from our audience. And we are very pleased to receive so many questions. So we dedicated some time to answer them. And I'll then start with the first one. We've been discussing the importance of circadian rhythms, but we also know that sleep is regulated by process S. How might sleep pressure influence or interact with what we come to understand about circadian rhythms?
I can go. I think it's really, I didn't show this, but I think they're just mirrors of each other. And it is totally, we're beginning to recognize more and more, whether it's at the molecular level, at the cellular level, or the systems level, that they affect each other. So we know that when you mutate, for example, mutation of the clock gene affects sleep homeostasis. But we're also beginning to recognize that when you sleep deprive individuals, for example, humans, you affect the level and also the timing of circadian clock gene expression. So so even at this very basic level, it is. And clearly when you affect, and I think Martha, you can talk about the hugging neurons and some of those things where sleep deprivation, how homeostatic sleep recovery is actually affecting, it's also shutting down the circadian clock neurons so that they permit one at the wrong circadian time to actually recover sleep. So
yeah, I can't add anything more to that. That's I think it's a very, very complicated issue, and you can also separate them if there are situations where the sleep homeostat becomes active too early or has too much sleep drive too early, and then you have this gap in the middle of the night where you're not sleeping and then the circadian clock comes in. I mean, there are all kinds of indications for why process S and C are hypothesized, and I think that there even is some data where there could be even a third component. Yeah, so
process Z, is that the process Z?
Oh, it would be appropriate, Phyllis. Yeah, so I have nothing more to add.
Thank you very much. Then we go to our next question. It's come from US. You have already mentioned a lot about future direction, but still, what do you envisage at the next question in terms of both basic and clinical approaches to circadian science?
I mean, that's I mean, I hope that Phyllis has her favorite clinical challenge. I think, I mean, I really do. I'm talking about circadian organization because I think it's really an incredibly complicated problem. We know so much about, these different levels, sort of as though we've sliced across the levels, the cell, something about the organ, a little bit about the organ, a lot about the organism, but we don't sort of see how they really work together. And I think that it's overdue that we understand this a bit better, and it will be very complicated because it's a bit of neurology, it's a bit of endocrinology, it's a bit of microbiology. It's a really complicated formula. So we have to identify sort of reduced complexity systems, I think, in order to start to address these questions. So circadian organization is, I think, sort of a threshold we need to get past to really be able to be effective at circadian medicine. I also think the way that chronotype changes with age, it systematically becomes later and later and later during adolescence. And then only, I think it's not really appreciated that it only gradually becomes earlier through adulthood. It takes longer to come back to quote unquote normal baseline than it did in adolescence. And I think that this volatile time of when your chronotype changes is super interesting. Probably mostly endocrinological, but maybe also a little bit developmental. And until we understand those, it's, well, we should not do circadian medicine, but we will do it better if we understand those variables.
Yeah, I totally agree, Martha, that the key here is, I think somebody who is Ray Silver said the issue is going to be in the tissues. And how then to be able to realize that, I do think we need biomarkers that reflect the function of the circadian organization, different levels, so that we can monitor those and see how they do interact, right? What is going up? What is going down? And I think we should put a lot of effort in looking at that, and that is both for the basic and clinical science. I don't think we can move forward without having some really, I think, good biomarkers. They may be specific necessarily, but they should reflect what's going on at the physiological level.
So I think there's a way, there's a path to start on that. That is, it's overwhelming to think of trying to monitor all those different organs, but there are some that tend to go together, that tend to move together or resynchronize together with jet lag and so on. And for a start, if we could just develop biomarkers that would track one of that group and one of that group and one of that group, then we could just do some screening and get a lot of very, very valuable fundamental data, I think, just to get an idea of what the space looks like.
Yes, and then we can validate that with interventions and see whether they respond.
Yes,
that's where the rubber hits the road, right? Do they really respond to that?
I mean, I think it's not so. It's of course huge and expensive, but it's not impossible, and it's not. It's imaginable. It is imaginable.
Amazing to see how many challenges we still have ahead of us. Huh? Gives again the big push to continue in these fields, definitely.
Yeah.
So we have a next question, and this is again a bit about the future, let's say. What advancements do you see on the horizon for making circadian phase assessments more practical and scalable for clinical use?
That I think Dr. Merrow mentioned that dim light melatonin onset may or may not be it may be a gold standard, but it can be quite impractical, especially under the conditions that it needs to be monitored in, which is dim light. I think the wearables and nearables that are emerging, the idea of being able to monitor multiple rhythms, as dirty as they may be, all at the same time, in real time, over many, many days, under various different conditions, and using machine learning to spit all this in and out. I think that probably will be where it's going to be scalable. It may not be as specific as we like, but I think we could be applying that. I think that's where it will, at least in part, should be heading in the clinic.
I think that's a great suggestion. I think that's probably the number one. That's where we really can run. I think also, I mean, you mentioned at the end education. I think the more we have scientists and clinicians aware of these things, the more timing will sort of somehow leach into more and more things. So, for instance, these horrible stories where drug trials fail by a tiny bit of a statistical percentage point. And if you only we think that if you would only give, I think there have been a couple of cases where you would give the drugs at a standard circadian time rather than randomly, that you would then achieve significance. I mean, I think that, once we sort of get a broad knowledge, then people pay attention to those things, then there might be some easy benefits that come back. I think that basic scientists are not accustomed to the process of getting things into the clinic also. And so some of us sort of have a hard time even imagining how that works and how what we do might be relevant, you know, and, you know, short of, you know, we would never, most of us would never think of even starting a company, for instance, but maybe you have to be thinking about those things to push things along.
Thank you very much. Maybe we'll take one more question. What are the barriers to using a translational approach with current rhythms? Is there anything that cannot be translated from the more basic research to the clinic? Or in other words, are there caveats that we need to keep at the front of our minds?
So I mean, the big caveat has been that circadian experiments are so hard to do because, you know, if you think about monitoring people over at least 24 hours, if not a week or a month to get your data, this has been just, and so on one hand, monitoring patients, on the other hand, doing the experiments, it's costly, it's time consuming. Our colleagues, when they, our non-circadian colleagues, when they review our grants, they look at how little we do and they can't imagine what we're dreaming of. But imagine doing a circadian experiment where you're monitoring something for days or weeks and then measuring all the values from that. Whereas if you have an experiment on some enzyme that you're purifying, you just do a one-time point and then you're done. So I think that that's a barrier to moving along. And you know, biomarkers, of course, that are validated, they're a wonderful solution to that, where you can take 1 or 2 time points, and then you have something that is hopefully reliable.
Thank you very much, Martha, and thank you very much, Phyllis. I think that we will close now our question and answer session. So we would like to thank you once again, Dr. Merrow and Dr. Z, for your brilliant talks and this fantastic, mind-blowing discussion. And thanks to everyone who joined us today. Yeah,
Thank you so much for having us, and thank you so much, Phyllis, for working on this with me.
Of course, my pleasure. Thank you.
It was great, yes. Thank you very much, and thank you all of our attendees as well that sent us the questions and will join us in the webinar. And
Thank
see
you.
you next season, right, Ludmila?
Yeah. See you next season. Have a nice day.
Thank you.